Hi guys, welcome to the second part of our lecture. Last time we ended with this slide right here. It was talking about the difference between chromatin and a chromosome. So chromatin was the unwound DNA found throughout interphase, and this is where DNA can be used. Remember, DNA is the code to make a protein. So the DNA in chromatin is easily accessed to be read and turned into a protein, which we're going to talk about that process in another chapter later on. But chromosomes over here, they are not easily accessed. They're wrapped really, really, really tightly around those proteins called histones. When they're wrapped really tightly like that, like you would wrap your Christmas lights to put them away in a box to organize them, it makes them easier to separate and make sure we get one copy of each chromosome in different cells when we go through mitosis and cytokinesis. Remember, mitosis is the division of the nuclei, so this is where the DNA will be divided during mitosis. So right before mitosis, we need to make sure that we wind that DNA really tightly into those packages like chromosomes and keep them really organized. But when they're wound up into chromosomes like this, and we're used to seeing chromosomes represented as this X, then the code cannot be read and proteins are not being made at this point because it's packed up too tightly. I also want to point out that this is actually one chromosome right here, but it's two copies. So this is one chromosome and this is the other chromosome. It's the exact same chromosome, so it might be chromosome number 1, but it's two copies of chromosome number 1. So chromosome number 1 and chromosome number 1. And we're going to talk about that more in a little bit. But that's what gives it its X shape. Okay, so moving on. When chromosomes, there are certain parts that are really, 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 really tightly wrapped. And those are called heterochromatin. They're very compact, and they're so compact and packed up in boxes that they're not transcribed. They're not made into proteins. This would be like if you were to move and you have all these boxes and you're unpacking the boxes that you're going to need and you're going to use, but some of the boxes you're not going to unpack. You're going to put them in the attic because you don't use those. This is what your cells do as well, because if you think about it, every single one of your cells has every piece of DNA, the code to make any single protein in your whole body. But your skin cells, they don't need to make the same proteins as your liver cells or your blood cells. They all make different proteins, but they have the code to make every single one of them. So each cell will have the DNA sections that it needs to make certain proteins that it needs will be left out. And those that are left out and more open and easily accessed, those are the euchromatin right here, EU chromatin. That means true chromatin, euchromatin. Those are going to be the ones that are not packed away in boxes. Now then, the proteins that you have this code for and you don't need, you're never going to make it because you're not a liver cell, you're a blood cell or whatever that you are. The ones that you will not need, they're inactive. Those are going to be wrapped really, really, really tightly around those histones and packed away in boxes in the attic because you don't need those. That's called heterochromatin. And then there's telomeres. Telomeres are going to be the very ends of those chromosomes. They're cap-like strands of DNA. They're very repetitive, and they used to be considered junk DNA, but we've actually learned a whole lot about telomeres in the last few years, actually, and it turns out that they're actually pretty important. They protect the chromosome because over time there's going to be a lot of damage to the chromosomes just from dividing and from just interactions that it has. And those telomeres are just the extra cap so that the pieces that are damaged are not the ones that are needed or required to make these proteins that we need. So what we find out is that telomere length is actually an indicator of age and overall health of an individual. Because as you get older, those telomeres are going to get shorter because they've been damaged. So a shorter length of telomeres, a shorter cap, is going to mean that the health is not as good. There's going to be more issues with that chromosome, so there's going to be more problems there. So it is an indication of overall health. There's a lot more research being done on telomeres right now, so you expect to see that in the news coming up as well. So we have the three pieces. We have telomeres, we have euchromatin, and we have heterochromatin. Those are all terms that you will need to know. Okay, let's look at mitosis and cytokinesis. And remember, these are both part of the mitotic phase, but they're two separate steps. Mitosis is going to be the division of the nucleus, so it's nuclear division. And cytokinesis is division of the cytoplasm, or of the whole entire cell. Before mitosis begins, the chromatin is going to condense. Remember, chromatin is that bowl of spaghetti, where all the DNA is spread out so it can easily be accessed to make proteins. Well, that's going to condense right before mitosis into those distinctly visible chromosomes that we're used to seeing like this. So these are chromosomes. There's chromosome number 1, and there are 2 copies of this chromosome number 1, because this right here is what we call a diploid individual. We're going to talk about that soon. It means that there are 2 copies of every chromosome. So here's 2 copies of chromosome 2, 2 copies of chromosome 3, 2 of 4, and each species has a characteristic number of chromosomes. For example, this roundworm here only has 4 chromosomes. Rice, 24 chromosomes. But it's not necessarily more complex. It's not a more complex individual depending on the chromosome number, because as you can see, humans have 46 chromosomes, but chimpanzees have 48, and dogs have 78. So that's all an evolutionary determination there. So. Okay, so I mentioned the term diploid. Diploid means that there are 2 sets of chromosomes of each type. So just like we saw right here, this right here is actually an array that we're going to talk about in a minute called a karyotype, and it shows each one of the chromosomes laid out by size. And this one's showing that there are 2 of chromosome 1, 2 of chromosome 2, 2 of chromosome 3, and so on. That's called a diploid individual. That happens because there will be 1 from the mother and 1 from the father, 1 copy from each parent. Humans have 23 different types of chromosomes, represented twice in each cell. So we have 23 from mom, 23 from dad, and that gives us a diploid number of 46. 23 from mom plus 23 from dad equals 46. So our diploid number, represented by 2n, is going to be 46. Now, the haploid number, if I ask you what the haploid number for a human would be, that's going to include only 1 set of chromosomes of each type, and that's going to be 23 for humans. Only in our body, every single cell in our body is diploid except for the sperm and the eggs. So our sperm and our eggs both have 23 chromosomes, and that's it. Okay. During interphase, a cell prepares for cell division. So organelles are duplicated, and each chromosome is going to be duplicated. And we're going to have some vocab words we're going to talk about here. So when the chromosomes are duplicated, they're going to consist of 2 identical double helical DNA molecules. So there's going to be something called sister chromatids formed. There are 2 strands of genetically identical chromosomes. So you can see right here. These are sister chromatids. We started out with this 1 blue copy, this chromosome 1 that we got from, let's say, dad. And this is chromosome 1 that we got from mom. After DNA replication, you're going to have 2 copies of the DNA 1 chromosome that you got from dad, and they're going to be joined together at a point called the centromere. And you're going to have 2 copies of the chromosome 1 that you got from mom joined together at the centromere. So we have sister chromatids holding hands. Whenever you think sister chromatids, think of kids, chromatids, kids holding hands. You don't want to put chromatin, remember that's different. Chromatin is that bowl of spaghetti. chromatids, looks like kids holding hands, and they're holding hands at the centromere. Okay, so these are chromatids. Now during mitosis, those centromeres are holding the sister chromatids together, and then sister chromatids are going to separate, and each becomes what's called a daughter chromosome. So when these sister chromatids let go and are not holding hands anymore, then they're called daughter chromosomes. Daughter chromosomes of each type are distributed to the opposite daughter nuclei. That way we end up with one of each in each cell. Just outside the nucleus, so remember we're inside the nucleus with the DNA. Inside of the nucleus, I don't have a picture here for you, but inside of the nucleus is where your DNA is that we've been talking about. But just outside of the nucleus is something called the centrosome. The centrosome is a microtubule, so it's like those Spider-Man spindles we used to talk about. It's going to organize all of that. And the centrosome contains two barrel-shaped centrioles, which are going to be like the refill cartridges for the Spider-Man shooting microtubules that we've been talking about. I showed you guys a video of the vesicle transportation where it looks like little ants carrying bubbles along the microtubules. It was kind of cool. That's the Spider-Man fiber that I was talking about. The centrosome is what's going to make these. So centrosomes organize fibers composed of microtubules, which allow the cell to change shape for cell division. And it also organizes the chromosomes. So this is going to be a big player in mitosis. Centrioles contain the fibers. So centrioles are these tubes right here inside of the centrosome. Okay, keep that in mind, and what we're going to do next is talk about the different phases of mitosis. I'm actually going to put that on a separate video that we will talk just about mitosis. So I will see you guys in a minute. Thank you, miss you.